Bismillahirrahmanirrahim. Genetic code. Lecture number 2 in the translational process. First of all, we know about the genetic code. Genetic code, it is basically the dictionary which translates the nitrogenous bases into a amino acid sequence in a growing polypeptide chain. The word or the present in the genetic code, it relates the nucleotide bases. So nucleotide bases, it form a word in the genetic code. Codons, it is the sequence of three nucleotides that are present in the messenger RNA, and there are 64 codons. Means all of these four types of nitrogenous bases, that is uracil, adenine, guanine, and cytosine, they combine in different ways so as to form the 64 types of codons. And then these codons, they are translated and into a specific amino acids sequence of amino acids in a polypeptide chain. So the genetic code, it provides a table to correlate the nucleotide sequence with the amino acid sequence in a polypeptide chain. Out of 64 different codons on the messenger RNA, 3 are the termination codon and 1 is the initiation codon. The termination codon UAG, UGA, and UAA, whenever it appears on the messenger RNA, it will lead to stop the translational process. While the AUG, it is present initially at each messenger RNA of eukaryotes and prokaryotes, and it incorporates the methionine. Basically, AUG is it codes for the methionine, and this AUG is present at the start of each messenger RNA. But when it is present inside of the messenger RNA, then it translates to the methionine. Characteristics of the genetic code. Genetic code, it is always specific, mean there is no any doubt to code this genetic code. It is always the code, the nitrogen sequence, and the codons which is present in the genetic code, it always translates to a specific amino acid, and this translation, it is always universal. So universality as well as specificity, they are the characteristics of the genetic code. However, there are only 20 standard amino acids, and for 20 standard amino acids, there are more than 20 codons present in the genetic code. So 61 codons present for 20 standard amino acids. It means that there are more than one codon for 20 standard amino acids. So degeneracy or redundant. This genetic code is redundant or degeneracy. And here, the genetic code, it is always read in this way. And there is no commas, there is no any punctuation, no overlapping. If we read in 5' to 3' direction, then AGC is first codon, UGG is the second codon, similarly AUA is the third, and CAU is the fourth one codon. There is no any punctuation, no any commas, no any full stop in the genetic code. It is always written in this form. This one is the genetic code. These nitrogenous bases, they form the start or the first letter of the codon. These nitrogenous bases, they form the second letter of the codon, and they form the third letter of the codon. And with the combination of these letters U, it forms the codon. Similarly, UCU, and similarly UAU, UGU. This one is the middle letter. At the top of these nitrogenous bases, they form the middle letter, start letter or first letter, and the third letter. And about 16 amino acids that start with A nitrogenous base. Similarly, 16 amino acids which contain U at the middle of their codon. And AUG, it is also a start or initial. It is the initial codon, start codon. And 3 are the stop codon. These 3, they are the stop codon. Here, the stop codon UAA, UAG, and UGA, while AUG is the um, initiation codon or the start codon, and others are like valine. Valine has such different codons. Alanine has such different codons. Similarly, threonine has also more than one codon. Proline also has more than one codon. Here, serine, leucine, phenylalanine, and glutamate, glycine, all of they contain more than one codon. Now, what happens when there is any change in the single nucleotide on the DNA, which also change the codon on the messenger RNA, which lead to the different type of mutation. If a single change of nitrogen base occur at the DNA level, it is known as the point mutation. This point mutation may be a silent mutation, it may be a missense mutation, it may be a nonsense mutation. A silent mutation is such that when there is a single change in the nitrogenous base take place on the DNA level, then it will lead to the incorporation of the same amino acid in the polypeptide chain. For example, TTC, it is the nitrogenous base, or these are the bases present on the DNA, then it codes the or it make the codon on the messenger RNA AAG that codes for lysine. But if there is a change in point mutation or there is a point mutation take place at C, and instead of C T is incorporated, then what happens? The codon will make this, the codon will be this one, and but this codon is also for lysine. So this change in point mutation, or when the point mutation take place in this level or at this stage, there is no change of the amino acid. This one is known as the silent mutation, mean mutation occur but it will not change the amino acid on the polypeptide chain. However, sometimes a nonsense mutation occur which form the termination codon. For example, this one, ATC. A is replaced by U at the messenger RNA because A, sorry, it is not replaced, but A instead of TTC, the T is replaced by A. When it happens, this one is the, you can say that the base of the nitrogen species present on the DNA. If this point mutation take place at C, then it will lead to the incorporation of lysine. If it will occur at the first nitrogenous base, this one T, instead of T, A is present, then it make the codon UAG, and UAG is the stop codon. So a truncated protein is formed, and this truncated due to this nonsense point mutation, because it creates the termination codon. Next is the missense mutation. Missense mutation may be acceptable, may be partially acceptable, or it may be unacceptable. Acceptable in such a way that the protein is formed. Here, missense mutation, there is incorporation of different amino acids. For example, at the middle level, instead of T, there is a C. TCC mutation take place in the at the DNA level that make the codon AGG in the messenger RNA and incorporation of the arginine. Or instead of C, G will be incorporated that also incorporate the threonine. So when this middle nitrogen base it is mutated then it will lead to the incorporation of different amino acids in the growing polypeptide chain. This one is known as the missense mutation, means different amino acid incorporation of a different amino acid through a missense point mutation take place. This is the different types of the missense mutations. Missense mutation may be acceptable, may be partially acceptable, or may be unacceptable. If it is acceptable, it means the uh, uh, polypeptide chain has the same physiological function, but electrophoretic analysis show that there is a different amino acid present in the polypeptide chain instead of the original amino acid present in the original polypeptide chain. Partially acceptable means some of the property is same with the native polypeptide chain, but there is change in the some of the physiological properties. Partially same with the native polypeptide chain. For example, hemoglobin S, which is formed, it can bind the oxygen, but at deoxygenated form, it will be precipitated. So, this one is the partially acceptable missense mutation, and unacceptable means totally different of the physiological function. Unacceptable, the hemoglobin M. One of the example is the formation of the hemoglobin M, which can't bind the oxygen. So it totally differ the physiological function. Another type of mutation is the trinucleotide repeat expansion. In this mutation, there is a repetition of trinucleotides in the DNA, which also code on the messenger RNA, and there is amplification of the this codon. The amplification of codon will result to the extra amino acid residues in the on the polypeptide chain. The example of this trinucleotide repeat expansion is the Huntington protein. Huntington protein, it is the repeat expansion of trinucleotides, these trinucleotides CAG. Repeat amplification of or tandem repeat of CAG codon, which give rise to the this amplification give rise to the extra glutamine residues in the polypeptide chain, and then thus the Huntington proteins are formed, and this Huntington protein lead to the Huntington diseases. These are the unstable proteins, and then they accumulate and thus the formation of the protein aggregates. And if these trinucleotide repeat expansion, this happen at the coding region, but if this trinucleotide repeat expansion take place at the untranslated region of the gene, then it means it will decrease the length, you can say length, because it will increase the untranslated region and decrease the coding region, and thus decrease the amount of protein which has been produced. These are the Huntington disease take place at the trinucleotide repeat expansion take place at the coding region, and thus give rise to the amplification of glutamine, and this amplification will lead to the proteins which are aggregated. And other examples of and if it occurs at the untranslated region, then less shortening of the amount of protein take place. Then comes the supply side mutation. If mutation occur at this type, then it means that it may increase the coding region or may decrease the coding region if there is a mutation at the introns level. And frame shift mutation means there is an addition or deletion of one or two nucleotides, which also change the reading frame. The best example is the cystic fibrosis, which is a pulmonary as well as the digestive disease. Here, the deletion of three nucleotides from the coding region of a gene takes place. And this deletion, there is a loss of phenylalanine at 508th position. So, this mutation will prevent the normal folding of this protein, and these CFTR, they are normally function in the colloid channel on the epithelial cell, and so there is a production of thick sticky secretions in the lungs and pancreas, which lead to the lung damage as well as the digestive deficiencies. Here is the addition or deletion of the nitrogenous bases. For example, UCA, it you can say code for serine residues, but with the deletion of C, the reading frame will be like this one, UCA. This C will be deleted, then CUA instead of the next codon CCU. CUA, that is for leucine, but this one properly for proline. Similarly, if one nuclear nitrogenous base will be added, then what happen? It also change the reading frame. UCA, here the incorporation of the U nitrogenous base, then the next codon will be UCC, which codes for serine instead of proline. So all of these different types of nitrogenous mutations that has been occur at gene level, which form the different coding region of the messenger RNA, maybe a point mutations, maybe trinucleotide repeat expansion, or it may be a frame shift mutation, addition or deletion of one or two nitrogenous bases.